Hello everyone, today's video will talk about genetic engineering, theories of evolution, and evidence for evolution for our Tuesday's test. So we'll get started with genetic engineering study question. The first one says, what is genetic engineering? What is a designer baby? So first off, genetic engineering is basically if someone were to change the genetic makeup of an organism and change it to a sequence of DNA that's not the same as it was before, we call that genetic engineering. And exactly how is genetic engineering done? So basically, if you look at this picture right here, this picture shows you a bacteria plasmid, which is the circular DNA within a bacteria. What you can do is that you can cut open the bacteria plasmid and get rid of a part of the plasmid, or even just leave that part hanging open, but you will insert another piece of DNA from a different organism, such as human DNA, for example. You can insert that into the bacteria plasmid. So now you have the old DNA and you have a new DNA in there. So this whole process can be called genetic engineering. So gene splicing is when you cut open the DNA, whether it's a circular DNA or a linear DNA. As long as you're cutting open the DNA, we can call that gene splicing. A recombinant DNA, this word right here, recombinant DNA, is when you are forming a DNA that recombines the old DNA and the newly inserted DNA that you want. So you can have this one right here, this circle right here is a recombinant DNA because you have the old and the new. What is the function of a restriction enzyme? Restriction enzyme is a type of enzyme that actually allows you to do the gene splicing. So when you want to cut open the DNA, there's restriction enzyme. So this restriction enzyme actually comes from bacteria and is used by bacteria to help it cut out viral DNA because virus insert its DNA into bacteria and bacteria doesn't want that. So bacteria naturally produce restriction enzymes, and people found out about restriction enzyme and we use it now to do genetic engineering and cut open DNA. What is the purpose of gel electrophoresis? So if you look at a picture of gel electrophoresis, you can have a DNA or RNA or protein, and if you want to separate the different fragments, let's say you cut open a whole bunch of DNA, you cut one DNA into 5 pieces, and you want to see how big each piece of the DNA is, you can run it through a gel and see the differences. So differences in sizes. So if you have a piece of DNA that's smaller in size, it's going to travel from the top to the bottom faster. And if you have a bigger piece of DNA, it's going to travel slower. So you'll have a band up top. So as an example, right here, you can run a whole bunch of DNA samples, A, B, C, and D, and what you can see is that A and C kind of have some pieces that are the same. This one and this one and this one are similar pieces of DNA, and this one are also similar pieces of DNA. So if someone were to ask you A, B, C, and D, which one is most closely related to each other, A and C is probably the one because we have the most amount of similar bands. We have 1, 2, and 3 bands of DNA that are exactly the same. That means those pieces of DNA are cut the same way. So that's just an introduction for gel electrophoresis. Let's see, is there another example that's good? All right, I guess if you can look at this picture right here, all 3 of these bands show up at the same place. That means those 3 bands are probably similar, similar lengths of DNA. The next one is, how is clone done? Okay, so if you look up a picture for clone, oh, this is not the right one, biology. All right, so let's look at this picture that we saw in class the other day. We have this sheep. What this sheep is doing is that it's donating the nucleus of a stem cell. So after we take out the nucleus from a cell from this sheep, we fuse it with an egg cell without the nucleus. So we have this sheep and we took out the nucleus, and now we fuse this nucleus from this sheep with the cell itself without an egg cell without a nucleus from this sheep, so that there are enzymes within this egg cell that can allow the cell to divide properly. So after this nucleus and the cell fuses, it divides into an embryo and then eventually turn into, and then we put this embryo into a carrier sheep so that the baby sheep, the cloned sheep, can grow up within this sheep, and then this little lamb is born as a clone of this original sheep. So what you need to know from this picture is kind of read it properly and interpret what's going on in here. We have a donor nucleus, and because this little sheep got the nucleus from this sheep, and DNA comes from the nucleus, so this sheep is a clone of this sheep, not this one or this one, because this one only produced the cell without the nucleus, and this sheep is the carrier of the baby sheep. Okay, what are some pros and cons of genetic engineering? You can look up a whole bunch of those. We also talk about those in class, so I'm not going to talk about those. All right, so this is the end of this video. The next video is going to talk about Darwin versus Lamarck.